At its core, issues of a clean environment is a matter of public health. May I now invite Dr. Ilavarasi, Assistant Professor of Zoology, to introduce the resource person of Technical Session 4, Dr. Saravanan Rangaraj, Research Officer, School of Natural Science, Banker University, United Kingdom.
Honorable dignitaries, respected teachers, and my dear participants, good afternoon. It's my great pleasure to introduce the chief guest of this session of the International Conference on Innovation in Biomedical Science Research. We are honored this day by the presence of Dr. Saravanan Rangaraj, Research Officer, Environment Center, Burns, School of Natural Sciences, Bangor University, United Kingdom, who is going to talk to us about the sensing technologies for environmental and human health monitoring. Sir did his bachelor and master degree in microbiology at Madurai Kamaraj University and PhD in chemistry from National University of Ireland. Sir has a versatile experience in research and his research interest is in biological fuel cells, biosensors, bioelectrochemistry, electrode surface engineering, and bioengineering. And Sir continued his research activities since 2005. For his contribution to research, Sir received several awards, including Charles Parsons Energy Research Award and Young Scientist Award. Sir, despite having spent almost his time in the research activities, with his dedication and commitment, Sir published no number of research papers in various international journals. Sir is a towering personality. Other than research activities, Sir also in the field of electrochemistry, microbial redox chemistry, molecular biology, microbiology, biochemical analysis, bioengineering, and instrumentation. On behalf of the management, organizing committee, and the participants, I welcome you, sir. Over the session to sir.
Thank you, ma'am. Am I audible?
Yes, you're audible, sir.
Yeah, okay. So I have some graphics in my slide. So if there's any problem, so feel free to interrupt me at any time.
Sure, sir.
Very good afternoon to everyone and good morning here. Hello.
Now it's okay, sir. We are able to see the presentation.
Yeah, okay. Yeah, can you see my presentation?
Yes, sure, sir. We are also able to hear you, sir.
Yeah. Yeah, sorry for the technical issues here. Is everything okay from your side?
Excuse me, sir? Okay, sir. Okay, sir.
Is that okay? I'm audible? Is my screen?
Yes, sir. Yes, sir. Yes, sir. Everything is fine.
Perfect. Very good afternoon to everyone. I'm very happy to be on board on the presentation side to present the topic on sensing technologies for environmental and human health monitoring. So I'm presenting here from Wales, Bangor, which is in the northwest of United Kingdom. You can see the pin here. So we have like 4 countries here: Wales, England, Scotland, and Northern Ireland, so which is the United Kingdom. And since everyone knows, since Jan 2021, UK is a separate country. It's no more in European Union. So we got separated from the European Union and we are in almost unique in European Union now. So Bangor has university has a very big campus spread out across the Bangor, which is, you can see the main building here, and this is our department, Environmental Center Wales. We call us ECW. The research we focus on is mostly on environmental side. We're looking into more of agriculture research, the pollution, and also we got projects on tracking COVID-19 in wastewaters. And the main important and main theme of our research is environmental and soil science. And my part of the research is to develop the sensor for soil monitoring. So this is very important, and it's a new prototype we are developing for agriculture research, which will be more of in use in providing a sustainable support for farmers in future. So in general, my my goal of my research is to take the agriculture research to next stage called precision agriculture using sensors. The goal of the talk is today is to focus on two areas. So one is environmental monitoring and human health. I'll be showing how the sensors will be used to monitor both the field, because both are interlinked. The human health is also depend on the environment, and environment is depends on the human as well. So both are interlinked. So once environment is spoiled, definitely human health is being also have issues. So and once the human health is spoiled, like for example COVID-19, so we're using a lot of masks and throwing a lot of garbages, and that environment first spoils the environment. So it's more of interlinked. So my research is mostly focused on researching on key issues that the current the world is facing on, and I have developed few sensors with our collaborators and with our colleagues that can monitor the environment and also sense the human health as well. So it's good to have a broad perspective of research. What I'm going to talk about, it will be more of general, how pollution is caused and what type of sensors can help to fix the pollution. And then also I worked on developing a human sensors for human health, like basically it's a biosensor, because I'm using a bioanalytes from the human body. And we have developed a few technologies that can help the human health as well. So I'm going to talk about my research, give a broad perspective, and then I'm going to show how the future looks with the system from now in next 20, 30 years. That will be the goal of the talk today. So since I give a basic introduction, what sensor? Sensor is just a physical, it can quantify any physical, chemical, or biological property. So it's a measurement objective. So that can convert any substance, any analyte or any target analyte to readable signals, such as electrical signal or light or luminescence or whatever may be. So the output information is more important, what we are trying to measure it and what the end user is trying to read, or end user is going to get benefit out of it. So there's different type of sensors. So the one sensors I'm more particularly interested is the electrochemical sensors. That's all the whole talk about. And this is my area of research. Being a microbiologist, since I had a huge transition, transforming myself from microbiologist to electrochemical electrochemistry side, and also I have touched few other subjects like physical chemistry, electrical engineering, and also surface chemistry, bioenergy conversion. And I'm dealing with a lot of biochemical engineering fields as well. So it's kind of a multidisciplinary project, and there's a lot of people involved in it. It's a nice journey from moving from microbiologist to touch all the diverse field, and that's all about the science. So you can't stick to one field. If you want to survive, if you want to learn, if you want to grow, you need to learn, keep learning things, and help growing for us and also for the society. So I'm going to talk about the sensor technology in environmental monitoring. So this is more of my current research, working more on agriculture and developing sensors for soil sensing, for nutrient management for agriculture. I want to start with a slide with the world population. So this triggers the environmental pollution. If you see the slides here, in 1950, the world population was like close to 2 billion. And in 2050, we expect around 10 billion in the world. This is rough idea. It could be more or less. And 1/3 of the population will be from China and India. And if you take India, if you see the area and the population density, it's going to be very challenging for everyone to tackle the environmental pollution. And the most important thing is the healthy food. So the well-being starts anything. So you can have a technology, you can have everything, but if you don't have proper healthy food, and you don't have a proper body, and you don't have a proper things to deal and survive in the world. So the well-being starts with a healthy food. So since the population is growing, and the agricultural land is being shrinking, India is an agricultural land, and most of us, we had a traditional agriculture practices that's been moving away. All the people who contributed for agriculture produce is a bit reluctant to do more of agriculture, and they're moving to other rest of the jobs, and poverty is being the the key issues of the farmers. It's really pathetic. So the basis of that, I recently came to know that in India, every year we are losing 30,000 hectares of agricultural land, and this is really alarming. So that gives more pressure on the agriculture. You to use agrochemicals, so plant growth promoters, fertilizers, and all the soil conditioner to produce the food in a short span of time with the high yields, the pesticides and indoor farming, which is a health risk for everyone. So the pollution starts from there and triggers everything. So from there, it triggers the anthropogenic activities, triggers the environmental pollution, and that affects the human health. So this is about how we're going to deal the system, and we're going to fix that's more important. So we have air, water, soil, and noise pollution. So this is more in general, like everyone is aware about the soil pollution, water pollution, air pollution. So soil, mostly chemical fertilizers from industry, or pesticides from agrochemicals, pathogens, or livestock excreta or sewage, water. This is the main thing that pollutes the soil. So in terms of water pollution, it's more of general agriculture and wastewater and heavy metals. And recently, it's antibiotic resistance. Antibiotic vaccines is going to be a huge problem because everyone takes the medication like with antibiotics. So once with the excreta and all the fecal waste that gets into the wastewater, all the bacteria is becoming more resistant. So one challenge example we're facing is that one example is the COVID-19. So it's hard to contain now. We are tackling it. And on the other hand, the air pollution, this is the most dangerous pollution, the greenhouse gas emissions and all the nitrogen dioxide emissions from the vehicles. So the methane, nitrous oxide, and all the carbon dioxide and chlorofluorocarbons are very challenging to contain. So on the effects on the human health, the environmental effects, and also economic effects. So the government has to spend more on containing this pollution, which is more challenging, and it's our responsibility to deal with to deal and fix the pollution from our side. It's our responsibility. So agriculture pollution is a serious threat to the world. As everyone knows, the farmers use a lot of fertilizers, so which is not the right thing. The land and soil has everything. If you do a proper farming, you can have a sustainable farming, and that gives you more healthy agriculture produce, and one can have. But instead, all the corporate companies or all the profit the companies looking towards a profit-oriented, they're pushing farmers or pushing people to use the chemicals, fertilizers to the soil. And what happens in next 20, 30 years, we won't be having a proper soil, and it's very hard for us to get a proper food and proper produce for our survival. Imagine for next 20, 30, 40 years, the shortage of agriculture produce of vegetables or water may be in high demand. So we might be put upon a rationing system that you have only a certain amount of food supply, certain amount of water supplies, you need to book online. So it will be the cases in future. So this is a time like our research is mainly focused on fixing the agriculture pollution. We're using the sensors, and we're developing few sensors, and we successfully tested it for since 3 years and 4 years, and now we're trying to validate it and trying to use and spread through the farming communities. So if you hear, imagine farmers supply fertilizer, so not necessarily. So if he does a proper testing, and he can know what exactly it is in soil and all the nutrients. So one key nutrient is nitrate, N, which is the basis for everything, for the amino acid for protein production in plants. So it has been thrown much more, and that goes off, runs and leaches into water, and that produces more nitrous oxide. So agriculture is one of the main causes of any pollution that's contributing equal to the other pollutions for the world, and this is very hard to contain as it will be more pressure. So as I said, all the wastewaters and all the agriculture chemicals are being washed away, and it's getting to the sea. And you can see these are the recent satellite from the NASA that's taken from, I think, somewhere close to the United States, and you can see the algal blooming due to the nutrient wash away. And once, imagine if the algal blooming covers the whole sea, and it will be a problem in the ecosystem, and everything will spoil. If the ecosystem is spoiled, you can see the dead fish floating, and affects the drinking water, greenhouse gas emissions, and we won't have a proper adequate water, clean water for our society. So eutrophication, the dead zones, is very harmful. So as the agriculture practice contribute to global air pollution, and the industry must adapt. So this is what we are trying to insist of how a system can be fixed and can be resolved at this stage. And if we don't do that, it will be a huge problem. So as I said, if you see the chart here, the agriculture is more of polluting compared to the industries, transport, buildings, and energy. It's 24% of the pollution comes from agriculture. It's really very hard to digest and hard to see what's happening here. We think it's a green field, but it's not a green field. It equally pollutes to the other pollutions, and agriculture is also one of the main contributors for greenhouse gas emission sector. So you can see the methane, nitrous oxide, and carbon dioxide. So three main gases, greenhouse gases emitted from the agriculture field, and this is due to the over application of fertilizers or nutrient or whatever may be. And also another problem is meat eating. So more people are being converted into vegetarian to non-vegetarian. I'm not blaming that, but since you have a choice of eating, the choice of eating meat is getting more, and animal rearing is more. It's a challenge as a pollution as being getting involved in triggering into the system and the society. So as a whole, what we are doing is to develop the soil sensors. This is I'm going to talk about the research what we're doing with the sensors here in Bangor University, and one of my the backgrounds. So I'm developing soil sensors, which could be a key to precision agriculture that can be optimized the fertilizer application and soil health monitoring. It can tell what's in the soil and what the nutrient, and the farmers can know what exactly it is. So here is one type of the sensors that can be deployed in the soil, and then that gives you continuously what type of nutrient. One example that we're doing is a nitrate as a urea. It tells you how much amount of urea is there. So once the sensors being deployed in the soil, so farmers know the health of the soil, and he can act upon based on the nutrients. You don't need to apply fertilizer all the time. You can save money. It saves the environment, and imagine how lot of carbon emissions producing from the fertilizers. So this one good thing where we're trying to do from the traditional system to converting that into more sophisticated system, fixing the agriculture pollution. So overall, the main objective of the project is to provide a farming support to farmers to feed a correct amount of nutrient required for the soil, protecting the soil, and also protecting the environment. And obviously, it will benefit the human health, producing good food. So this is kind of a mapping we will generate in future, like where exactly the nutrient deficiency and where exactly the nutrients more excess, and you can throw the fertilizer off. So the future, how the future in the agriculture industry looks in the current scenarios, that most people will be moving away from the agriculture practices, agriculture farming, and all the artificial intelligence and the sensors will be the game changer of the system, where the sensors will be, the drones will be using to spread the fertilizers or throw the chemicals that can sense the field, and you can also diagnose the disease. And they can monitor the crop yield, and smart data is like where the farmers have everything in hand, and based on the field. Water stress is more important, like these sensors can more specific, they can reduce the water application and conserve more water for the rest of the world, and field monitoring and the variable rate of fertilizer and soil erosion. So as a general, the sensors will be of a central nervous system for agriculture system in getting more produce and less with the less pollution. So this is one concept we are trying to develop and see as a future, and we got a lot of data for 3-4 years, and we're trying to validate the sensor data, and that will definitely benefit the farming. One interesting thing I would just to show here, and everybody will be very interested to see what's happening. So the Bill Gates is the largest landowner of United States. So this is very interesting for me, and I want to share with everyone. So the man behind the computer, the revolution is the computer is getting into the farming. So he thinks the farming is the more important thing, and I think definitely the farming with the good hands, with the people like Bill Gates will secure clean environment in future. So he has acquired like 268,984 acres in United States, and he's going to help farming develop systems, use the Microsoft Azure. He has developed a Microsoft Azure as like a Microsoft Windows. So the Microsoft Azure will help the farmers to make it more sustainable farming. So the one thing he clearly believes that the landowners and farmers are the backbone of everything, and he's going to provide all the innovative support and sustainable farming to all the people. And we are into the race in such systems and to give a hand for all this technology revolution. And that's all about the environment. And I'm going to show what's going to happen with the human health with the sensor technology. As a pollution, as we don't have a proper healthy, so we have a lot of problems with the human health, and it's important to have a check, not necessarily every time, but at least if you have something online or something that gives you an indication what exactly happening in the body, it will be more useful. I want to talk about the body sensors. So before going into man-made sensors, I want to talk about the body sensors, which is more really, we have innate sensors in our body mechanism. So it really is amazing and beautiful. You can smell, taste, hear, see, and touch. So these are the 5 sensors that body can do. So basically, a body is a sensor. As example, skin, you can touch temperature and feel the pain. Tongue can realize the chemicals or the taste. The nose, you can sense the chemical gas or something. The eye can see, and ear can listen to sound and all the things. So all controlled by the receptors or sense organs and handled by all the message being traveled through the neurons and all the central nervous system process, and we can sense what's happening. So the body is of a sensor. But what happened? But there are certain things beyond the senses, beyond the body mechanism could sense. For example, the body fluids and biological functions, yearly detection of disease, and monitoring electrical signals. So these can't be monitored by the body, and you need some devices. So that's where the sensors can play a major role for the diagnostics. So that's a one good thing anyone can see in future. The sensors will be playing an important role. That's beyond the human sensing. So beyond the natural sensing, sensors can play a good role. So here's a chart how the sensors can play a role of monitoring the health status and then the reaction to the external factors. So this is more spontaneous and it can respond. So for example, you do a daily activity and you don't know what's happening with the body, and it's hard to predict. So whether sensors can predict what's happening and they can detect any of abnormal things happening in the body, and then it can lead to diagnosis and support. It can inform the health issues well before the diagnosis and the treatments. So this is one thing, the basic concept of the sensors are looking at. If you look at Europe and US, there's a lot of research is carrying out with the sensing and developing biosensors, and this is one of the biggest research innovation that will be coming in the next 10, 20 years for the future. So one good thing is that Clark is the father of the biosensor. So he's a man who he invented an oxygen electrode. So it's called a Clark oxygen electrode. So that it monitors the real-time oxygen of the blood of the patient's blood, and it made surgery safe. So millions of people around the world, the medical field is being benefited out of such innovation of monitoring the oxygen levels. So this is one example of how the electrochemical sensors revolutionized the world. So basically, the sensors what it can integrate the biological element with the transducer and produce electrical signal in a single analyte to convert the signal into detector. So I want to show how the sensor looks. So it looks exactly similar to the human body mechanism. So I showed you the previous slide. So you see you have the receptors and central nervous system and all the motor neurons, and it processes and the brain sensors, and we can respond to it, right? The same thing, the sensor also has kind of receptors. So it can analyze a sample, it can detect the enzymes, antibodies, nucleic acids, proteins, anything it can be sensed, and that can convert. They can use a basic transducer electrical system as a central nervous system, and then finally it displays what exactly the substance is or target element is. So the biosensors being a promising for medical devices, it can do the rapid diagnostics, which can lead more timely and effective treatments. The one good example I can quote is the blood glucose meter, where most people are using it for the diabetic. You have a small, you just prick a blood and have a small chip, and that reads the blood glucose. So that's the one innovation that transformed the whole world. So you have the lab-based home lab at home, and you don't need to go to a hospital every time to do a blood check or check the glucose in the lab. So it's just more simple. It gives you a basic indication how your body blood glucose is. And the futuristic approach, what it can mostly, these sensors will be used for detecting the cancer biomarker, so it can detect cancer very early. So these electrochemical sensors are more sensitive and accurate, and it can transform the world. So I want to show how the electrodes function, and this is a basic schematic mechanism, how the sensors will be looking into it. If you see an enzyme, for example, glucose oxidase, you can convert the glucose into gluconolactone as a byproduct, and then it converts with the oxygen, converts hydrogen peroxide. So we immobilize these enzymes onto the electrode, and that can sense the glucose. So since the protein, the enzyme is a protein, is covered by the protein, the active sites are not talking to the electrode. So we use certain mediators to trap the electrons from the oxidized substance to the electrode surface. So this is how the mechanism we used on the electrode devices. It looks simple, but if you see the mechanism behind it, it's more complicated. And the beauty of science is more amazing to see how it works. So this small strip has the electrodes on an enzyme immobilized on it. And once you put the glucose, the blood that has a glucose, and that gets you the reading into as an electrical signal. So this is the basis of what the glucose monitoring devices everyone has. It's very affordable, and it's very it's more of a normally charged, and it took a lot of research for several years to bring out the technology. So this is one innovation of the sensing field. So as I said, electrochemical sensors is going to be very important, that can detect the enzymes, antibody, aptamers, and everything. So you have a system here. Basically, it has a carbon electrode with the connections here, and you can target any substance, any analyte like enzymes, protein, antibody, DNA, and once you confirm the recognition binding, you have an indicator target analyte that can sense the signal. So with the electrochemical signal response, you can see how well the bio conjugation happens, and this gives an indication. So if you see the application of biosensors is huge. It can monitor the soil, it can monitor the environment, disease detection, water monitoring, toxin detection, drug discovery, pathogen discovery, and food quality monitoring. So it has a versatile application with all the sensing, not only the biomedical, it can be extended to all the field as well. So one interesting thing I would show you what will happen in future is the self-monitoring of their own health. It's mostly wearable and semi-implantable devices. As you know, most people have a smartwatch and also smart devices that can tell you how much calories you burn, and also what the heart rate, the blood heart rate, and you can also see the oxygen levels of the physical body while you do the exercise or normal day-to-day activities. So this is one example, but they are not biologically confined target analytics. And once if you want to do the physical analysis, you need to have some kind of a biorecognition system, the biosensors coming in, and where you can monitor your cortisol, your blood glucose based on sweat. That's everything. It will be since having a patch of sensors as a tag that to monitor. It's a non-needle, so you don't need to take any of the sounds. So blend with the sweat. Basically, it analyzes the basic thing: your salt, your cortisol. Cortisol is one of the hormone that tells you how stressful you are. That can control and keep control and keep an eye on your stress levels. That's one good innovation. there'll be in future. What these wearable sensors sense, the tear in your eyes, it can be, future could be a lens, artificial and intelligent lens. You can have a sensor based saliva based, and you can also have a sensors with a breath based system. And the more important thing is implantable sensors. I'll be showing how this will happen in future, and also interstitial fluid body sensors as well. So this will be very interesting. It looks like a Hollywood movie, but it's going to be the one of the basic thing in future if you want to have a proper health. So it's how the wearable sensor looks will make a diagnostic. As I said before, it's a wearable technology, but it's not actually wearable technology. Something behind science behind that can trigger your body mechanism. So these sensors can be patched on the skin, so that can track down your the sweat. You can track the glucose levels, cortisols, and all these salt concentrations of your body. And also, it can be semi-implantable. So these called the microneedle sensors. So it looks sharp here, but it is not that sharp. So it can just pierce on your skin as a tattoo. It's called a microneedle patch. And also, once this is done, it can start to read your interstitial fluid that's collected on the devices and keeps the monitoring. And also, it could be a semi-implantable devices. Such system, the biologic system, can live for like few months for any a particular post-operation analysis. This can help to see how the body mechanism helps. So this is kind of a general view how the sensors will be looking in future. And one example I have to show when I was in University of Connecticut. We collaborated with James F. Rusling. So his lab was doing electrochemical detection methods for cancer detection. So this is really one of the nice innovations. So they have some experimental proof that the prostate-specific antigen that's responsible for the prostate cancer can be detected early as 5 years before the clinical signs of disease is being known outside. So this kind of point of care diagnostics or any of advanced sensing system where you can predict what will happen in future, that's really very a good thing to have as a platform instead of getting diagnosed and spending lakhs and lakhs for the hospital, and act properly well before the diagnosis. So it's less invasive, and treatment protocols are very easy once you have the blood samples and you can detect the cancer. So here's I don't want to go much more into detail. So here's the signals, the electrochemical signals based on the concentration. So you can monitor the picogram levels per milligram and milliliter of the concentration, very minute of the proteins, cancer proteins that can give us a signal. So at this stage, if someone gets a treatment, it can prevent the cancer. So this chart shows you how once the humus triggers in the body, it takes some time, and these sensors can detect well before the prediction diagnosis and before getting treated, and you save your money, and also you can save your life as well. So this is one good example how these things will look. And also some research group in Finland, they're looking into I want to tell that I had a good collaboration with James F. Rusling. Good to learn about the sensing, incorporating all the electrode system, and we had a nice collaborations with this lab. And also some of the companies in Finland universities looking about how tracking the brain disease. So the sensors could monitor the effectiveness of brain treatments. As I said, the neurotransmitters, more people are being affected by Alzheimer's and Parkinson's and schizophrenia and epilepsy. So this will be a problem if you don't monitor properly. So this the group of research group from Finland, they came up with devices, a demonstration that can track your neurotransmitters and keep track of how well is your body mechanism is. So they can predict what's happening with your neurotransmitters. So these neurotransmitters are the brain sensing hormones that can help to improve your brain function and controls the body mechanism, the central nervous system. So this is another example. And lastly, the recent innovation is that point of care testing for COVID-19 in a test tube using in less than 30 seconds. So you don't need to have, if such systems develop research innovation, can minimize the PCR sampling or all the extensive lab analysis. So the University of Nevada in US, they have recently demonstrated a screen printed electrode sensing. They have synthesized and prepared the antigen COVID-19 virus with the two domains, and they have immobilized on the sensor side with the more functionalized surface, and that can they prove that it can recognize the COVID-19 virus in the sample. And they did a lab demonstration, and now they are trying to validate the sensors with the actual blood patients with the viral transport medium. So one of such system is more beautiful and it's very rapid. Once you prepare the surfaces for a specific antigen or whatever maybe, and this can be on-spot analysis. Say, for example, if you go to an airport or something, you just take your swab samples and make a solution, and you drop on the electrode. You know in 30 seconds what's happening. So it's a positive or negative, so you can move on. So you don't need to quarantine, you don't need to wait for like 7 days or something for the testing. So such system will be a kind of a technology revolution and innovation in the future. So these are the few things I want to show you from our research side, and also a general perspective. So as a key messages, what in general I want to talk about from all this thing is that if you want to have a proper healthy activity, you need to have a proper healthy food. So sustainable farming is the only way that to secure the food security in the future. Why I'm talking about food security is that it's very important. Food security is an adequate supply of food to everyone at all the stage. Imagine for next generations, our kids or whatever may be, next generation will be face a huge problem with the pollution and the food scarcity. And we need to fix, and everyone has to think about the environment and how the future food security is going to be. So personally, I prefer like everyone has to get involved in farming, and they can do the one, how they can develop the own art of growing own vegetables as much as possible, not depending upon, so you know what's happened, what is there in it, and try to help the society as well. And also we need a plan to combat the environmental pollution, to eradicate all the problems in future. So if you take care of the pollution, if everyone takes care of the environment, like disposing of garbage and everything in a proper way, you can reduce the burden. So the government, whatever the government brings a plan, it's also a responsibility for every individual to take care of the environmental in future. So that's more important. So the science and technology progress, how benefit is the humankind. So I have shown you all the technologies, right? Science is doing its job, and people are developing few technologies, and some technologies are more benefiting, and some are basically a commercial oriented backed up devices. So it's very important for any regulators, for any technology regulators, that before getting into the people, you need to analyze and make it more affordable for the people and help the science reaching to getting implemented for the society. One good example is that you might have heard about the couple of brand new flights, the Boeing flight accident that killed 348 people. The Boeing 737 is the safest plane in the world until now, until there was some fancy sensor technology was incorporated, and that made that resulted in a crash in Thailand, and there's one more in South Africa. So this is one good example, like too much sensing technologies, where it also be a risk, a danger to humankind. So it's very important, the sensor technology, whatever we develop, it's more reasonable and more benefit to the people. Finally, I want to just give a note. It's not a suggestion, it's just a sharing. If you want to have a well-balanced life, stay happy. That's more important thing. I know it's very hard, but if you try to keep you happy, that's more the first thing of your healthy life. And eat healthy. Try to not minimize all your junk food. Try more vegetables, and that's more important. No rush. I know people are rushing up, people are running behind, not taking care of time on personal health and family, friends. And also, more important thing is to take care of your environment and health. So this is more important. It's very important to take it to the next level, and what we're going to leave for the next generation, that's more important. And more importantly, connect with people. So if you connect with people, share with friends, and this eradicates most of the mental problem, like a psychological problem, and think about the future and how the world will look into it. With that, I would thank you everyone for their time for listening my presentation, and thank the Holy Cross for organizing such a wonderful seminar and delivering a environment. I'm being as a part of it to talk, and I hope I have delivered some kind of useful content, kind of a broad perspective from our research side and the futuristic approach. So I thank the biotechnology for funding banking support project. Thank the professor and PA of the project, Professor Davy Jones, for helping us in all the implementation of this project. Thank you very much. I'm open to questions. I'm happy to take up all your questions. Thank you.
Thank you, sir. Now, the session is open for discussion. Participants can unmute and ask your questions.
Hello.
Sir, I have one question. That is, as India is concerned, how far we can implement this agriculture sensors in India, like whether Indian farmers can use such sensors?
Yeah, I think it's possible. So it's all about the policy makers, I say. So the government has to back up the farming. In my personal view, the farmer, whatever the technology you develop, I say 90% of the farmers won't be having a finance support to implement such technologies. So only 10% of the farmers, rich farmers, can implement such technologies. So I think it's a government and the policymaker responsible to disseminate all the technologies, which can benefit the society, and that's where the key thing to support the farmers. Imagine like farmers get like hardly like a few thousands in money for the profit, and they are working hard to produce the food for us. But what we are going to provide for them is to back up and kind of a support system. So that has to come from the government. We have a lot of Internet of Things. We have cloud. We have a lot of technologies coming out, and I can see what's happening here in the Western world, and that definitely Indian farmers can get all the technologies if the government supports. So that's my personal view.
Good afternoon, sir. So here we have few questions. First question, what are the different sensors used for soil monitoring?
Yeah, so we have sensors for checking the soil temperature, soil moisture, that tells you how much the soil is wet, whether the land needs water or not. And we are developing the nitrogen, phosphorus, and potassium, like NPK sensors. That's the main for the nutrient fertilizer sensors, and also sensors for, this is for the below ground I'm talking about. And for the above ground, we have the sensors for monitoring the pest, how well is the plants are, and we have the sensors for monitoring the chlorophyll content of the plant, that tells you how green, how healthy your plant is. That's one the sensors we have. And also we have the drones now that can fly over and just see how what is the nutrient deficiency with the plants. So we have everything in for the sensor. But one thing where the farming is lacking is the sensors to monitor the nutrient. So that's where we're developing a sensors. It's the first of its kind proof of kind of technology we're developing for the farming system. So this kind of sensors will be added to all the electrical other existing technologies to support the system.
Is there any adverse effect on wearing these biosensors for a long period of time, sir?
Yeah, I think definitely yes. That's why it's like they can benefit at some point. They also have a problems with the response. Sometimes the body what I can see, if this gets implanted, you have kind of an antibody produced and it thinks as an antigen. Sometimes you have kind of an allergic reaction. So it's also could trigger adverse effect. So that's why I said, like, whatever the technology we develop, we need to have more of validation before getting into the practical approach. And definitely, we need to be more careful, not affecting the human system for the sensing. I think this is possible. Like we can solve the problem.
Can we use these biosensors for pest control, sir?
So, sorry.
Can these biosensors play a role in pest control?
Yes, exactly. So the one good thing is that, as I said, like the above ground monitoring technology, like these sensors has some kind of the cameras that can detect the pest. So once you handheld devices or the drone devices that flies over, that can detect the pest. So recently I read an article that the few companies are developing a system where exactly you can predict see the pest within the plants. So it's possible, yes.
A few more questions, sir.
Okay.
Is the biosensors available to detect genetic disorder?
Might be in future, because the sensors, they're working on the DNA sensors. Probably gene editing will also be possible in future, but I can all the research in very basic level, like the protein, like the antibodies, immobilization. There are certain groups they're working on DNA sensors as well, having a single stranded DNA onto the electrode surface, and once your sample is being exposed to it, and you have the DNA which can hybrid for the target analyte, it can do is possible for the gene editing, and it can also it uses kind of recognition on it. But I think it's more of a PCR based for gene sequencing is more now, but it possibly we can also do the gene editing with the sensing technology in future.
Are there any biosensors commercially available which can be installed in a residence to monitor air pollution, sir?
As to my knowledge, I don't have those technologies will be used in the market, maybe only in the research side. So what I think in next 5-10 years, all the research which is sitting in the lab will be developed, definitely will be into the society, into the field, helping common man. As far as my knowledge, there's no such biosensors. There's sensors that can monitor carbon dioxide or nitrogen dioxide, CO2 levels. You can buy these devices available in the market, so that can sense the gas. So these gas sensors, they can monitor the air pollution. So it's more affordable. It's not too expensive, but you can have that in your home if you do a little bit search. But if you talk about the biosensing for the environment, I don't think so. We have such devices at the moment, as far as my knowledge.
Final question, sir.
Yeah.
How nanopore technology is working in biosensor?
Yeah, so this is a good question. So nanotechnology is really very recent field. So instead of having the electrode surface, so these nanoparticles, it will increase more of the sensitivity of the target analyte. So they're more specific, they're more efficient. So these, instead of using a normal carbon electrode surface, these nanotechnologies like carbon nanotubes or nano things that can increase your surface area, which improves the detection system, basically. So definitely nanotechnology, like people are using graphene, highly surfaced, functionalized surface, carbon nanotubes, functionalized electrodes, which is going to be also will be included in such technologies, will be used for more of a precise or a fast response, and definitely it will be helpful for incorporating all the functionalized nanotechnology onto the sensor devices.
Thank you so much, sir, for addressing all our queries.
Thank you. Thank you.
Once again, thank you, sir, for making us understand about the types of biosensors, its application and uses. And I'm sure that in the future, as sir mentioned, India will also make policies by that all our farmers will take up this biosensors and we equally compete with the agriculture trade market around the world.
Thank you, sir. Thank you so much, sir.
Thank you. Thank you. Thank you. Thank you.
Thank you, sir. Thank you so much, sir.
Genes are like the story, and DNA is the language that the story is written in. May I now invite Dr. Avila Jolie, Assistant Professor of Zoology, to introduce the resource person of Technical Session 5, Dr. Prashanti Namburi, Postdoctoral Scientist, Department of Ophthalmology, Hebrew University Medical Center, Jerusalem, Israel.
Respected dignitaries, cherished faculty members, and most valued participants. Good afternoon to all. I'm really privileged to have the pleasant task of introducing the speaker of this technical session 5, Dr. Prashanti Namburi, postdoctoral scientist, Department of Ophthalmology, Hebrew University, Jerusalem, Israel, who is going to talk about identification of genes that are associated with various ocular diseases. Ma'am is illustrious academician and excellent researcher. She has pursued her post-graduation in biotechnology in University of Madras and her doctorate in Madurai Kamaraja University in biomedical science. Her area of interest and research expertise are human molecular genetics, genetic variation and diseases, molecular biology, and gene expression. To her credit, Ma'am has received several national and international fellowships like Senior Research Fellowship from CSIR, Government of India, AFER Developing Eye Researcher Fellowship to attend annual conference in Florida, USA, and IERG Travel Fellowship. Her excellency in research is highly commendable, and she has published 14 papers in reputed journals with cumulative impact factor of 50. Dr. Prashanti is distinct with her unblemished oratory skills and presented several papers in international conferences. Ma'am postdoctoral studies focused on molecular genetics of inherited bacterial disease, and her findings was published in American Journal of Human Genetics, which carries impact factor of 10.8. Apart from all her achievements, Dr. Prashanti is humble, down-to-earth person, and my wonderful friend. On behalf of the Department of Zoology, I thank her for accepting our invitation and marking her presence here. I welcome you, ma'am. Now I invite her to enlighten our participants with her scientific ideas. Thank you. The session is now over to Prashanti.
Thank you, ma'am, for introducing the resource person. Participants, kindly post your queries of the session in the chat box, which will be consolidated at the end of the session. Ma'am, we request you to kindly take over the session.
Ma'am, you're not audible.
Prashanti, kindly unmute your audio. Prashanti, we cannot be able to hear you.
Ma'am, kindly unmute your audio, ma'am. No, ma'am, you are not audible.
Hello, can you hear me now?
Ma'am, you are
Can you
audible
hear
now.
me now? Yeah,
Yes, ma'am.
Yes, ma'am. Yes, ma'am. Yes,
I'm
ma'am.
sorry for that.
No, ma'am.
Can you see my slides? Are they clear?
Yes, ma'am. It is visible now.
Good afternoon to everyone. First of all, I would like to thank the organizing committee for this wonderful opportunity, and I would like to uh, give a special thanks to my close friend Avila for a nice introduction. So before getting into the topic, as you all know that I'm going to talk on identification of genes associated with various ocular diseases. This work was carried out at Professor Sharon Lab, Hebrew University. I'm going to split up today's topic as two stories. One is identification of centrosomal protein CEP78 gene, and the second story is on PRDM13 and complementary factor H gene roles on maculopathy. A small introduction about a human eye and how it functions. Cornea is the outermost layer that protects the eye, and there is a pupil behind it. When the light enters onto the eye, it enters through the pupil and then pass on to the iris. Iris followed by the lens. On the lens, the imaging will take place. So behind the lens, you can see a vitreous humor that gives a shape to the eyeball. So
Prashanti,
when you see, yes,
Prashanti, your
yes,
slide is not moving, Prashanti. Kindly move your slides, Prashanti. It is entitled.
are you sure? Because
Now it is okay. Now it is okay.
so you want me to start from the introduction?
You can continue now.
Okay, so a small introduction about the eye. The cornea is the outermost layer. When we see a vision, the light enters into the cornea and then followed by the pupil through then to the lens. And behind the lens, you have a vitreous humor that gives a shape to the eyeball. And from lens, the image will be sent to the retinal layer, which is the posterior most layer, and through the retinal layer, the signal will be sent through optic nerve to the brain. So electric impulse will be sent to the brain, and the brain, with the help of vision impulse, will send the signal. That is what which we see. So I can say in a human eye, the retina is the most important part. And today I'm going to talk about retinal diseases, how we identify genes that are causing various retinal diseases. And among them, the one which I'm going to talk is cones and rods. Those are the inner segment retinal layers. As you can see on the panel B, these are the rods, and then in a cone shape, these are the cones. And there are three types of cones. The function of these cones is to give a daylight vision and also the color vision to us, like the colors we see. And the rods, this will help in night vision. Suppose when we enter from a bright light to a dark room, then rods will play a role on it. So here on the right side panel, you can see the structure of rods and cones, the nucleus and mitochondrial and other material. On the right side panel, you can see there are rods. This is the electron microscope image, and then followed by the cones. As you can see, the cones is cone shape. In inherited retinal diseases, retinitis pigmentosa is the most common form with the frequency of 34.7%. So what is retinitis pigmentosa? Here comes the role of rods and cones. Whereas in retinitis pigmentosa, rods will get affected more. That means they get damaged. So the night vision will gone, and then followed by the cones. So in the beginning, the night vision will be lost, and followed by slowly the day vision will be lost. Prevalence of this disease condition is 1 in 5,000 in worldwide population, whereas the ethnic group that I studied, it was 1 in 2,200. So all these studies were carried out in an ethnic group of Jewish and Palestinian community, and vice versa of this eye condition. That means more of cones get damaged first, and later the rods. That condition is called cone rod degeneration. The prevalence of this condition is 1 in 40,000 people. Case of cone and rod degeneration, first the day vision and the color vision will be affected, followed by the night vision. So we have a cohort of 1,540 inheritable retinal diseases cases, and among them around 750 cases were solved. Two of the stories I'm going to share how we solved. And as I said, first story is CEP78 gene. This was published in American Journal of Human Molecular Genetics. So the study started by recruiting 6 probands from 5 different families. All these families from a Oriental Jewish ethnicity. That means Jews who are lived in Iran, Iraq, and Afghanistan background. So if you see each one, we call it as a pedigree. Here there are 5 pedigrees, and the round symbol is
Excuse
a female.
me, ma'am.
Yes.
Ma'am, please kindly change your slide, ma'am.
I did. You couldn't see. Okay, can you operate in, because I did change the slide. 1 second.
Ma'am, now the phenotype.
Yeah, this is what I'm talking about.
Fine, ma'am. Thank you, ma'am.
Maybe can I make it like this so that you guys can see more clearly? Now you are in phenotype slide?
Yes, ma'am.
Okay. So each one is one of the pedigree, and the round symbols represents the females, and the square symbols represents the males, and then the filled symbols are the affected cases, and the red arrow represents the probands for these families. And in these families, the problem is, as I said earlier, they have a cone rod degeneration, and also along with that, they have sensory hearing problems. They have vision problem and the sensory hearing problem. Did the slide change? I went to the next slide.
No, ma'am. No, ma'am, it didn't change, ma'am.
Can someone make a change there? Is it possible when I say next? Because for me, like, it has changed. I'm sorry for this inconvenience.
Ma'am, did you present your entire screen, ma'am?
Yes. I'm in imaging of individuals. Is it there now?
Prashanti, can I present your presentation, Prashanti?
Yeah. I will say next slide. You can go to the next slide, Avila. That must be the good idea.
Okay, Prashanti, then now you stop presenting so that I can present it.
Yeah. So I'm on imaging of individuals now.
Yes. Just you stop your presentation so that I can present it from here.
Okay, then how will okay. Can I
in
say now?
In a new tab, you can open it. You stop this, and in new tab, you can open your presentation and you can continue your speech.
Okay. Okay, now. Hello. Hello. Hello.
Yes,
can you hear?
Yes, ma'am, we can hear you.
Yeah. So coming to the imaging of individuals, on the left side panel, you can see the fundus image on topmost layer on the left side panel, normal individual where the hello.
Ma'am, I think, ma'am, you must stop presenting. Then only we can present the screen here. Ma'am, please stop presenting.
Oh my God. Okay.
Ma'am, you can go to the Google
Yeah.
Meet and you can.
Yeah, I did a stop. Hello, Avila. Hello.
Hello, Prashanti.
Yeah, I'm at 7th slide. Can you display that
Yes,
for me?
yes.
Am I running out of time?
No,
How
no,
much time?
Prashanti. You can do for the 45 minutes more.
Okay. So can I start with the 7th slide?
Yes, Prashanti, you can start.
Yeah, this is the imaging said that they all have a vision problems. So this shows what kind of phenotypic features they have. On the left side top panel, normal individual, you see that fovea macula are very clear, and the left side panel represents that fovea, macula, and other retinal layers which got damaged. And on the right side panel, FAF imaging, where you see normal individual is very clear, and when it comes to the patients, you can see a black color and white color spots, which are called salt pepper formation. So their phenotypically they had problems. That's why they had vision loss. Next slide. Next slide explains the audiometry. Because I said they also have a sensory hearing problems. So on the for normal individual, you can see the frequency of audiometry as a clear graph as a horizontal line, whereas in other individuals there is a fluctuation. So it says that they all have a sensory problem. Next slide, please. Next slide. So now, as I said, 5 families we have recruited with such conditions. So we started a study with family 679, whereas 2 of the brothers are affected and the parents are consanguinely married. They have a relation marriage. So in case of such marriages, always it's a good idea to go with a homozygosity mapping, because their genetic material is coming from the common ancestor. So we performed the homozygosity mapping on the symbol, as you can see, represented in with a red arrow. And then once we had a homozygosity mapping data, we mapped the data, and down panel you can see that mapping data for that patient, and on x-axis you can see the chromosome number, and at chromosome 9 you can see the red line, and also whereas at chromosome 19 you can see a black line which has a scale, higher scale, that represents that on chromosome 9 around 75 megabase pair and chromosome 19 around 85 megabase pair region had passed the criteria. So that means there is some high homozygosity region were located in those 2 regions. So when we look into the variants that were presented in these 2 locations, around 650 genes were presented on chromosome 9. Around 700 genes were located on chromosome 19. And these genes, when we look into the literature, none of them were harbored to the reported inheritable retinal diseases genes. Next slide, please. So we kept this data as one side, and then we performed the whole exome sequencing on the proband's elder brother. So when we perform a whole exome sequencing data, we used the HiSeq platform by Illumina, and then mapped using human sequence 19, and then we used the lab pipelines to filter the variants. And on the left side, you can see that whole exome sequencing data in a form of ANOVAR file. And you can see each ash color line is one read. So Each one represents one read, and then bottom line you can see the nucleotides. You can see that the nucleotide G has been replaced in green color with adenine. So glycine at one particular point of at one particular place has been replaced by adenine. So we compared this whole exome sequencing data with homozygosity mapping data. Can you go to the next slide, please? So when we, sorry, the previous one. Yeah, when we compared it, then we had come up with that both the siblings are sharing around 19 megabase pair region, whereas you can see on the location of CEP78 on chromosome 9, the region that has been shared by the two siblings were represented in a red line. So this was shared. When we look in detail into that region, we found out around 30,000 variants were located in that particular place. Can you go to the next slide? So from this 30 variants, how we picked the correct variant that is associated with the disease. There are many filtering methods which we have as our lab, which our lab has. So using that, we filtered and we have come up with 15 variants. Among those 15 variants, again by minor allele frequency, generally the variants that has less than 0.05 minor allele frequency are significant. So as you can see, MAP value with the oval circle, those three are more significant. And again, based on prediction tools like SIFT, polyphin, and mutation score, from these three, we figured out that you can see in a red highlighted one, CEP78 one is the one which is which may has an effect to cause a disease in this family, which may, because generally when we look into the mutations that occurred on splice site regions will have more pathogenicity towards the diseases. So we took this variant. Can you go to the next slide? And then we located where this particular variant is located on the genome. It was located on the CEP78 gene at exon 7. So we designed the primers for that particular region and did a Sanger sequencing for the available family members. So as you can see, Sanger sequencing data, the first panel, homozygosity wild type, it was done on unaffected sister of the family, and the glycine was as it is. And whereas the mother, who is a carrier, she is a heterozygous for glycine. Whereas in case of the proband, he has a homozygous variant. Glycine has been changed to adenine. So this is the cause for the disease in this family. So the same mutation we looked into the other families. As you can see in the MOL 773, the proband is also like M1 bar M1 represents that mutation with homozygous condition. So she is also carrying the same mutation. So in these two families, the same mutation is the root for the disease occurrence. Next slide, please. And then coming to this family, you can see that M1 at the proband M1 bar M2. That means the mutation only, the first mutation identified with the heterozygous condition. When we look into the entire gene by Sanger sequencing, then we found the second mutation, which is exon 4. Again, it is a frame shift mutation. And so these two mutations, we looked into the other families, and in the two more families, we found out the mutation 2 with the homozygous condition. As you can see, mutation 2, it's like a deletion of thymine nucleotide, which produces a again a frame shift mutation. Next slide, please. Yes. So now, among 5 families from the same ethnic background, we found 2 different frame shift mutations on CEP78 gene. Generally, when a population migrated from a bigger population to a small region, there will be a founder effect. That means they all will have a particular common genetic makeup that could be the cause for the disease. So, with the help of haplotyping analysis, we can identify if they have a founder effect. This is the data of haplotype analysis. As you can see in the red box, that those two mutations, the polymorphism that we looked, are appeared commonly in all the patients. So, which strongly says that this this changes has been occurred due to founder mutation. Next slide, please. So up to now, we identified this mutation at a DNA level. So we wanted to know that the same thing, like it has an effect at RNA level. Then only we can say that it has a defect in the protein, and then that can be associated with the disease. So what we did, we extracted the mRNA from the blood leukocytes for available sample, and then you can see on the gel, you can see on the gel like sample 1, lane 1, which represents that the defected short fragment. That means that they have a deletion of one of the exon, and lane 2, which is a normal individual. So they have the 290 fragment. So which in the normal case, they have a sequence continuously, whereas in case of the defect, the patient, there is a deletion of exon 7, and it has been confirmed by Sanger sequencing. You can see like the chromatogram, which represents that a dash line, that near that dash line, the exon 7 has been deleted. Next slide, please. Can I go to, yeah. So, yes, now we know that at mRNA level also these mutations are having an effect. We wanted to know how this gene is expressed in various tissues in different human tissues. So from like bone marrow, spleen, and different tissues, we again extracted the mRNA, and see, as you can see the panel on the left side, first and second, most of all the tissues are, in all the tissues, the CEP78 has gene is expressed. And down on the bottom, you can see PGM1, it serves as a housekeeping gene. On the right panel, you can see CEP78 is encoding 3 different transcripts. Very nicely, all the three transcripts has expressed with a similar expression level. You can see retina, it is a faint, and whereas thymus have a little higher expression. Next slide, please. So RNA-seq data from a human retina, I mean, you can see in the graph that it also confirms that all the 3 different transcripts are having same expression pattern. Almost the score, if you see, it has 3.12 and little less. So the down one, which is a schematic representation of entire CEP78 transcripts. And as I said earlier, one of the mutation was located on exon 7, and the second one on exon 7, which we highlighted with the pink color lines. Next slide, please. Yes. So we saw the mutation effect at mRNA level. Now we wanted to know that how this gene is behaving in mouse retina, also in the ear, because all the patients are having sensory problems. So what we did, we extracted the mRNA from the mouse retina at a different age points, like at the age of embryo 14 and postnatal 0, postnatal 30, and also inner ear postnatal 30. As you can see, when the embryo is developing, the expression is getting increased. on the left side panel, CEP78 expression at embryo level, little low, and uh, uh, when it is getting progress, you can see higher expression. And PGK1 is the internal control for inner ear sample. So it has the same housekeeping gene. It will have a same expression level at all points. And next slide, please. So now this slide represents that where the CEP78 is located in the retina and how it is behaving. On the left side schematic represented, I already showed you the cones and rods. The cones and rods. And next to that, it is called, it is a immunohistochemistry imaging of human retina. From bottom, like you can see green color rod. I'm sorry, I couldn't point out. And then there are some red dots. So here the green color represents CEP78 antibody and the red color represents rhodopsin, which is a housekeeping gene. You can see that it is a inner segment photoreceptor layer. The color is expressed in cones very well, which means that CEP78 is located in the cones. So cones, if get degenerated, then there will be a defect of CEP78. So because of CEP78 defect, the degeneration of cones will occur. So that can be clearly explained in this slide. Next slide, please. Okay, this is the comparison between human retina and mouse retina. As you can see that in the last bottom layer, all the cones have a good expression, fluorescent green color in the cones. So which clearly confirms that if CEP78 defects plays a role in cone rod degeneration. Next slide, please. So to conclude, the first story that we know in this disease condition, CEP78 gene is the cause for the disease. And also one of our previous lab member, they identified from the same family, centrosomal protein family, CEP250, associated with inheritable retinal diseases, one of the inheritable retinal diseases. So that means all these are forming as a network and causing a disease. Next slide, please. So these results were published, as I said, American Journal of Human Molecular Genetics. One of our collaborators also found simultaneously in mutations in the same gene with the same phenotype, which strongly suggests that in the different ethnic groups, CEP78 is the gene is the cause for cone rod degeneration disease. So moving on to the second story. Yeah, next slide, please. Do I have time, right? So
yes, Prashanti.
Yeah, okay, thank you. So this is identification of PRDM13 and complementary factor H genes role in maculopathy. So what is maculopathy? Again, maculopathy is a condition where, can we go to the next slide? Yeah. Maculopathy is like it is the center part of the retina where high concentration of cones will appear. So on the right side panel in the fundus imaging, you can see those are the macular regions where they got damaged. So like in all these patients had a maculopathy problem. On the left side panel, this study started. This study we have done it in one of the family, 4-generation family. As I said earlier, like the squares are represents the males and the round represents the females, and the filled symbols are affected cases, and you can see a bar, those are diseased cases, and there is on the first generation there you can see one circle with lines. Yeah, she has a mild phenotypic features, whereas other family members, they had a clear maculopathy problem. Only she is having mild phenotypic features. And there is no correlation between the age and the severity, because as you see in the fourth generation, the younger one, she also have high phenotypic features. Next slide, please. So in this family, we started performing a whole exome sequencing analysis on two of the people. One is on the, can you move on to the previous slide, please? Yeah, because as you see the inheritance pattern, this is autosomal dominant. So it's we have done a whole exome sequencing on the proband. That means a girl at fourth generation and her father who is unaffected. So when we are targeting for any mutations, you should identify them in a heterozygous condition, because it is an autosomal resistant inheritance pattern. Next slide. So we performed the whole exome sequencing on the proband, the girl and her father, and then we have come up with around 90,000 variants, and those 90,000 variants were identified together in the father and the daughter. So we have, first thing, how from these variants how to filter step by step analysis. Because it is a heterozygous condition, you should see only into the daughter and not into the father. So that's how we filtered some of them, and then we came up with the 3,000 variants. Again, from the 3,000 variants, as I said earlier, lab pipeline filtering methods, and then SIFT values and minor allele frequency, all this has been followed. And then one of the variant, which is a complementary factor gene, which already known by literature that this has an effect towards maculopathy. So we just sticked on to this complementary factor gene, and then as you can see on the left side, whole exome sequence data as a NOR file. You can see the black lines, those are the deleted regions, clearly represents. So clearly represents in some of the reads that some nucleotides has been deleted. This was confirmed again by Sanger sequencing. On the right side panel, you can see the chromatogram clearly represents the top one is a homozygous wild type where CTTAAG, and these are the 6 nucleotides has been have got deleted with the heterozygous condition. Down panel, which shows that those got deleted and with the heterozygous conditions. So this variation is leading to a deletion of 2 amino acids at 750 and 751 position, the leucine and the lysine. These 2 amino acids are getting deleted. So once we know that in the proband, this is the these are the amino acids getting deleted, we should know the similar pattern has been occurred in other patients or not. So we have performed the Sanger sequencing in all the other infected cases, and as you can see, I think you can see the we mentioned that M1 bar plus represents the wild type condition. M1 on the segregation analysis right side panel, please. Yeah, you can see M1 bar plus represents that wild type, and then M1 represents the mutation is carrying. Because it is a heterozygous condition, we have to represent in that way. So in all the patients, it is representing very well. But on generation 1, where the great grandmother, she has mild features, she is still having a wild type allele. So we have to answer why she is not carrying the mutation, though she has a phenotypic features, but with mild conditions. So keeping this in mind, we have moved on to the next level, because we should prove that this is the cause for the disease. Next slide, please. So we went into the literature and looked for the conservation status for these 2 amino acids. They are highly conserved. That means if there is any change in these 2 amino acids, that is going to affect functionally onto the protein, which can be a cause for the disease. So if you see a protein level, a downside panel, complementary factor H gene encodes 20 functional domains. Among those 20 functional domains, can you see the round circles 12 and 3? These are the domain 12 and domain 13. The two amino acid leucine and lysine were located the linker region on these two domains. So when there is a link between domain 12 and domain 13, the connection amino acids has been deleted. That means the connection has been lost. So which can be a major thing. Move to the next slide. So we wanted to see that does it making a role in a structural way. So this was using a Rosetta as a model. We built a model on A and B panels, top layer purple color. You can see a thin line above the green. Yeah, A panel. Yeah, that region is missing when it comes to the B panel. So this is the linker between two different functional domains. This is changing the property of the protein, which could be the cause for the disease. So we had come up with this result. And at the same time, interestingly, we were keep on reading onto the literature. Next slide, please. And surprisingly, two papers by Small et al. Group has come that a new gene has been identified in maculopathy. Macular dystrophy, that means which has a similar phenotypic features of maculopathy. So these two papers changed our entire direction from complementary factor gene to different direction. Can we go to the next slide, please? So what from their data, what they say is, in case of macular degeneration cases, a new gene PRDM13, which was located on chromosome 16, upstream of this particular gene has a few variants. They have identified few variants. These variants are the cause for the disease. So then immediately we started looking into their approach, and then we started, we have designed the genes that covers the region which they are suggesting that could, the region which they are saying as a hotspot region for the cause of the disease. So we designed the primers, and as you can see, the red arrow, homozygous wild type on the left side panel. Yes, the blue lines, which DHS on the DHS upstream, you can see there are three blue lines. Those are the variants reported by the small group. And then when we look into the same region, we identified a new variant, which is a transversion variant, adenine to cytosine. As you can see that in unaffected, it is in a wild condition, and heterozygous mutant at the bottom panel, mol 1154322, adenine has been replaced by cytosine. So then we again did a segregation analysis, and this particular variant is beautifully segregating very well in all the family members, including the great grandmother who is having a mild type features. So this we are considering it as a mutation 2. So in this family, mutation 1 and mutation 2 together are causing a disease. You can ask why? Because both the phenotypes, one is maculopathy and another one is a macular degeneration. Both are having similar phenotypic features. So both genes, they are located in two different chromosomes, yet they are in combine, forming causing a disease in this family. Since we already know from the literature, complementary factor H gene has a role in maculopathy, and PRDM13 from small group says that it has a role in macular degeneration. We stick on to the conclusion, and we don't need to go for the functional studies. And we could come up with the conclusion that these two are combinedly causing the disease. To conclude, can we go to the next slide, please? Yeah. two identified variants are segregated very well, and then we also looked for these two variants in other individuals who who are healthy, and they none of them are carrying. Only the patients who are having same phenotype are having are carrying these two variants. So in case of genetic disorders, it's always good to look into the phenotypic data and genotypic data to come to a conclusion. So in such cases, it's a